In, in the previous session, we learned about accommodation of DNA into the cell through the formation of chromosome, which is the histone protein. In today's session, we'll learn about the aspects of genetic engineering, mainly speaking about isolation of total DNA from bacteria. A gene is a unit of hereditary material. A gene sequence encodes for a protein or RNA which participates in various other cellular processes like metabolism, gene regulation, and other areas. The need for gene manipulation in vitro paved its way in early 1970s. With the subsequent development of genetic transformation in E. coli, the cutting and joining of DNA molecules and monitoring these two processes. These two processes are carried out by the restriction nucleases and ligases, which are the two main pillars of genetic engineering. These two enzymes will be dealt in detail in the further videos. There are 5 main steps in the process of genetic manipulations. That is, isolation and characterization of DNA fragments by various methods based on the requirement, obtaining the desired length of fragment coding for a particular gene by using restriction nucleases, which cuts the DNA fragment for desired length by recognizing a particular pattern in the DNA sequences. The ligases stitch these fragments with desired vectors. A detailed account on how restriction nucleases recognize particular pattern in the DNA sequence and how ligases actually stitch the fragments of DNA and in how many ways these two enzymes work in combination will be dealt in detail in the further videos. The next procedure being the insertion of these fragments into a suitable vector, transferring these vectors to suitable host organisms to obtain desired number of copies, expression of these DNA fragments, which is achieved by transcription and translation. Isolation and characterization of DNA fragments by various methods based on the requirement. There is various types of DNA. For example, if you take a bacterial cell, there is the regular bacterial chromosome. If present, there are plasmids. If the bacterial genome is infected by a phage molecule, there is also the phage DNA. Different procedures are employed for extraction of different type of DNA. So, in this video, we'll be talking about preparation of total cell DNA. The total cell DNA is the genomic DNA content present in an organism, may be bacteria, animals, or plants. This includes plasmids also because it is representing the overall DNA content of an organism. The plasmid DNA only specifies the extrachromosomal DNA that is independent from the bacterial chromosome, whereas the phage DNA or bacterial phage DNA represents the DNA from the phage genome. Phage is a bacterial virus which infects bacteria and it injects its DNA into the bacteria and it uses the bacterial replication machineries to undergo replication. This genome is called the phage genome, which is isolated by using special techniques. Preparation of total cell DNA. This procedure needed for total cell DNA preparation is divided into the following steps. The culture of bacteria, which is grown and harvested. The cells are broken to release their content. This cell extract is treated to remove all the components except DNA. The resulting DNA solution is concentrated. Culturing of bacteria. Bacteria are generally cultured in liquid medium for genetic material extraction. There are 2 media generally used to culture bacteria, the M9 media and the LB media. M9 media is a definite media which consists of all the components required for culturing of bacteria, whereas the LB media is an indefinite media which does not contain all the requirements but is sufficient in providing certain nutrients for the growth of bacteria. The next steps involved in the isolation are preparation of cell extract, purification of DNA from the cell extract, concentration of DNA, and measurement of DNA concentration. Preparation of cell extract. Cell extract is prepared using two chemicals, the EDTA and the lysozyme. EDTA and the lysozyme is used in combination to prepare the cellular extract, which these chemicals in combination disrupts the cell membrane and cell wall and liberates the cellular extracts. This is subjected to centrifugation to separate them to DNA, RNA and protein, and the lysed cells settle in the bottom of the pellet, forming precipitate. The next step is the purification of DNA from the cell extract. Two methods are used to purify DNA. Removing the contaminants by organic reagents and enzyme digestion. The standard removal of contaminants is by addition of phenol and chloroform in 1:1 ratio. This mixture precipitates the protein but leaves the DNA and RNA in the aqueous solution. If the proteins are present in large amount, proteases are used to break the polypeptides to smaller subunits, thus rendering this mixture accessible to phenol-chloroform mixture. Another analytical technique used for isolation is the ion exchange chromatography for the purification of DNA. DNA and RNA are both negatively charged, including some of the proteins. These require positively charged resin for its binding. The separation is based on the concentration of the salt. As the electric attachment is disrupted by salt, removal is more tightly bound. Molecule requires high concentration of salts. Higher concentrations of salt is required to remove DNA, hence DNA is precipitated at last and is eluted with more salt content. The next aspect is the concentration of DNA. This is achieved by ethanol precipitation. This is the technique used to obtain concentrated DNA. This procedure is carried out in the presence of sodium salts at -20 degrees Celsius. Absolute ethanol precipitates the polymeric nucleic acids. DNA is pulled out in this way. Another advantage of using ethanol precipitation is that it leaves behind the short chains of polynucleotide, that is the RNA, that are completely eliminated in this step, giving a purified DNA. The next step being measurement of DNA concentration. DNA concentration is measured using UV absorbance spectrometry. The amount of DNA is directly proportional to the UV radiation absorbed by the solution of DNA. The absorbance is measured at 260 nanometer, at which the wavelength and absorbance corresponds to 50 mg of double-stranded DNA per ml. This is also used to attest purity of DNA, where the ratio of absorbances at 260 and 280 nanometer is measured to be 1.8. Ratios less than this indicate the presence of contaminants such as phenol or protein in the sample. The diagram here represents the overview of isolation of total DNA. Some of the steps, like the fifth step in this diagram, represents using a mixture of phenol, chloroform and isoamyl alcohol in a definite ratio. This is a developed method which depends on the material taken into consideration for extraction. Sometimes the material may not be bacteria. This can be employed for the extraction of other materials like the plants or animals. So, modifications are made in the technique to accommodate the extraction of particular DNA. Thank you for watching this video. Please do like, share and subscribe to our channel. To know more about the isolation of total DNA, please do check the link in the description. Thank you.